At this point, I'd like to take a break from the main video and just run some problems. I'm going to make a template strand of DNA. We're going to randomly generate a template strand of DNA, and then I want to give you guys an opportunity to try to solve this for yourself. I want you to tell me what the coding strand is going to be, the strand of DNA opposite to that original DNA strand, and what the mRNA would look like as well. That is, what would RNA polymerase make based on this template strand that we are going to produce? And then finally, what would the polypeptide look like at the end? So here's what we're going to do. I'm going to randomly generate a DNA strand using this four-sided die. It's got 4 points on this pyramid, so each point is given a number. Whichever one is sticking up is going to be the number that we do. For our purposes, I'm going to say that 1 equals an adenine, 2 equals a thymine, 3 equals a cytosine, and 4 equals a guanine, A, T, C, and G. So let me just create this random number here. Okay, that's a 1, so that is an adenine. That is a 2, that's a thymine. That's a 4, that is a guanine. 1 is an adenine. Okay, so there's our DNA template strand here. Now, across from this template strand, there's another strand of DNA. That's the coding strand. And you know the base pairing rules. A binds to T, and T binds to A. C binds to G, and G binds to C. So using those base pairing rules, see if you can figure out what the coding strand would look like. Go ahead and pause the video. I'll wait a few seconds, and then I will give the answer. All right, so across from adenine, we're going to have a thymine. Across from thymine, we'll have an adenine. Across from guanine, we'll have a cytosine. This next triplet is going to be much, much easier. It's just cytosine, cytosine, cytosine. And then up here we have guanine, guanine, guanine. Up here we have guanine across from this cytosine. We have adenine across from this thymine. And then we have thymine across from that adenine. So that's what the coding strand of DNA would look like. Now the mRNA that would come from this template strand follows similar base pairing rules, but in RNA, whenever you see an A, there's going to be a U hydrogen bonded to it. Whenever you see a C, it's a G. Whenever you see a G, it's a C. Whenever you see a U, it's an A. So we're going to make this mRNA molecule based off of the template strand. We have an A here, so the mRNA would put a U in that position. We have a T here, same as before, we put an A there. We have a G here, that's a C, 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 G, 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 and G, A, U at the end, GAU at the end. So that's what our mRNA sequence would look like. Now notice, if you have any U's in this polynucleotide, you know automatically that we are looking at a strand of RNA because there are no U's in DNA. Up here, if you see T's in a polynucleotide strand, then you know automatically that it is DNA. If you were to see a strand that looked like this, like that, with T's and U's at the same time, you immediately know that that is wrong. That can't exist, okay? We never have T's and U's in the same polynucleotide strand. So, the base pairing rules, there are 5 nucleotide bases. They are T, A, G, C, and U. These ones up here are DNA, and these ones down here are for RNA. All right. Now, we have the polynucleotide here. We have the mRNA molecule, the messenger RNA. So now what we need is our genetic code sheet. So here is our genetic code sheet, and we need to take a look at these codons. Every triplet of RNA bases is called a codon. So we have 4 codons here. 4 codons to translate out. The first one is UAC. Now, you should take a moment to open up this genetic code document from your slides, or take a look at it in the textbook. There's a copy of it in the textbook chapter, and see if you can translate what UAC stands for. And then try to translate these other ones as well. Pause the video, and I'll wait a little bit before I give you the answer. Okay, so there's 4 codons here. There's going to end up being 4 amino acids at the end. So each 3 mRNA letters gives you 1 amino acid. UAC is our first codon, and if I come up to this chart, in the upper left-hand corner, we have the U's, and then the entire column across the top row starts with U, and then I see the second letter across the top, A, is in this third column here. So we're going to zoom in on UAC, and we see that UAC gives us tyrosine. So TYR. This is not 3 different chemicals here like we had here, right? This is not 3 different nucleotides. This is just an abbreviation for that amino acid tyrosine. Okay, there's 1 amino acid in this position. CCC. And then the second column has my CCs, so I will zoom in on that second column here. CCC is proline. CCC is proline. No matter what letter we had for the third nucleotide, it was always going to be proline. And this is something that's very robust about the genetic code. Remember, there are 64 combinations of these nucleotide letters, but there's only 20 different amino acids to actually choose from. Therefore, some of these codons are going to be synonyms, right? If you ever heard that story that, Inuits have 150 words for snow or something like that, sometimes we have words, 2 different words that mean the exact same thing. And this is true of the nucleic acids and the polypeptides as well. They have synonyms in this language. And typically, it's the third base in the codon that doesn't matter so much. If you have the first 2 letters of the codon, you can usually take a pretty good guess about what amino acid is going to be coded for in that codon. The reason has to do with nitty-gritty chemical reasons related to the transfer RNAs, which we'll talk about in a second here. All right, third codon, GGG. G's are across the bottom row, and GG is in the final column, so we're talking bottom right, and any combination that begins with GG, regardless of what the third letter is, you're going to get glycine. So the third amino acid is glycine. Okay. Final codon, we have GAU. GAU, oh, I can just move around like this. GAU, if we come in here, we can see that there's 2 different codons that code for aspartic acid, and one of them is GAU. So ASP is our final amino acid here. So what did we do here? We took template DNA, the sequence of randomly generated nucleotides, and we replicated that DNA. We created a compatible coding strand. So when we did that, we were doing the same job that DNA polymerase does inside your cells. Then we took that template DNA and we did the job that RNA polymerase does. This is transcription, where we were copying down the information into an RNA message. Now, one thing you might want to notice here is that TAC up here on the coding strand, UAC down here on the mRNA, CCC, CCC. So the reason why the coding strand is called the coding strand is because it resembles the mRNA. It looks like how the mRNA will look. The only difference is that whenever you would have a T on the coding strand, you instead get a U on the mRNA. I would suggest when you do these kind of problems that you do the coding strand and the mRNA separately, and then you confirm that they resemble each other at the end to make sure you didn't make any mistakes. It's a good way to check your work. Then we took that mRNA message and we did what the ribosomes do. We translated it out into these 4 amino acids. Now let's do a second one of these. We're going to do the same thing. Randomly generate a DNA strand. Template. DNA. 2, that was a T. Okay, so we have our template DNA here. What would the coding strand look like? Coding DNA. What would the mRNA look like? And what would the polypeptide look like? Right. Solve all 3 of these on your own, and then I'll come back with the answer. So pause the video now, and then I will give you the answer afterward. All right, so A to T, C to G, so we have A, G, A up here on the coding strand. We have A, T, A for the second one, G, 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 and T, G, A. Those are our nucleotides for the coding strand. mRNA, we're going to, again, copy from this template strand using our RNA base pairing rules. So I see a T, that means I'm going to put an A. I'm going to guess that some of you put a U there for your first letter. It's okay, it's a common mistake to make, but make sure you write this understanding in your head. Whenever you see the T on the RNA, you're going to put an A. Whenever you see an A, that's when you would put a U down here. And that's why it's good to do both of these separately and then check to make sure that they look the same. Whenever there's a C, there's a G, that's always true. Whenever you see a T, you put an A. A, U, A for this next one. G, 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 and then T, G, A. Oh, I'm sorry. Even I make little errors periodically, but it's always good to have a check. Now, these are our codons, our 4 mRNA codons that we are taking a look at. And up here, we have the coding DNA. So AGA, AGA, good. ATA, AUA, good. GGG, GGG, good. TGA, UGA. All right, we are good to go on this. Then we want to translate it to a polypeptide. So AGA, let's go to our chart up here. AGA, where are you? We have the A's down here on the 3rd row, and then the AG's are way over on the right-hand column. So here's our AG's. AGA is arginine. Arginine. Okay, what's next? AUA. AUA is probably around here somewhere, isn't it? It's probably just over to the left. Where are you at? AUA. There you are. Isoleucine. ILE is the abbreviation for isoleucine. Make that a. Ooh, I'll make that a little bit more clearly an L. ILE here. Okay, so 3rd, GGG. I think we had this one on the previous problem, but the G's, all the codons that start with G are down here on the bottom row, and then the GG's are down here in the bottom right-hand corner. GGG is glycine. Is glycine. And then UGA is a stop codon. UGA is a stop codon. So I know I made a little bit of a mess here. Let me just rewrite that. Our polypeptide sequence would be arginine, isoleucine, glycine followed by a stop codon. Okay? And the stop codon just means that the sequence is terminated. Even if there were still some codons going after this, if there was like A, U going on after this, if you hit a stop codon, translation stops immediately. So this is all that would be translated from this point.